In a previous video, we looked at the tally of ATP produced when glucose is completely oxidized into CO2. And we noticed that at this point here, the two NADH that are made via glycolysis and that are in the cytoplasm give us this strange result here. They either lead to the full 5, which is the normal 2.5 ATP per NADH, or it gives us 3, so 1.5 ATP per NADH. And when we read here, it says it depends upon the mechanism used to shuttle the NADH equivalents from the cytosol to the mitochondrial matrix. So that's what we're going to look at in this video here. What are the shuttles that cells can use to move NADH electrons from the cytoplasm to the matrix? And this is called the cytosolic redox balance. So balancing the NADH in the cytoplasm and the matrix. And there's two ways that cells can do this. And some cell types use one way, and some cell types use a different shuttle. And so when we look at this here between 3 or 5, it depends upon what cell type we're looking at. All right. So let's concentrate on the glycerol 3-phosphate shuttle first, because that's the one that's a little bit easier to understand. And what it depends upon is a enzyme here in the inner membrane of the mitochondria that takes the electrons from glycerol 3-phosphate and transfers them to Q. Okay, so thus bypassing. So those electrons from NADH in the cytoplasm then normally would end up in complex 1 here, right, where we get those 4 protons being pumped. But we're missing that. Those electrons, and we'll see how we get them to glycerol 3-phosphate in a second, are going to go to Q. So we're going to lose some potential energy of that NADH in this type of shuttle. So here's a better look at it. And here is the cytoplasm side, or the cytosol side on the top, and then here's the matrix side on the bottom. And glycolysis produces our NADH, and now to shuttle those NADH, to shuttle those electrons into the electron transport chain, the way it works is there is a cytosolic glycerol 3-phosphate dehydrogenase. This is an enzyme that we've seen before. I haven't had you memorize it yet, but it is what converts glycerol 3-phosphate into dihydroxyacetone phosphate, getting glycerol to the glycolytic pathway to either make glucose or break it down to pyruvate. Now we're using this enzyme here in the opposite direction. We're using it to take dihydroxyacetone phosphate, using it to oxidize NADH in the cytoplasm. Okay, so it's taking the electrons from NADH and thus becoming glycerol 3-phosphate. Glycerol 3-phosphate passes through the outer membrane of the mitochondria, gets to the inner membrane, and there is another enzyme here, the mitochondrial glycerol 3-phosphate dehydrogenase. That then reoxidizes glycerol back into dihydroxyacetone phosphate, and in so doing, reduces Q. And then Q is good to go to complex 3, be involved in the Q cycle, and so on and so forth. Now, what that means then is that instead of each of these NADHs being worth 2.5 ATPs, they're bypassing complex 1, just like succinate, for example. And so instead, that NADH is going to, those two NADHs are each going to give rise to 1.5 ATP per NADH. Now, these the cells that tend to do this type of pathway are skeletal muscle and the brain. Now, the malate aspartate shuttle is a little bit more complex, and it takes advantage of those electrons riding on the citric acid cycle intermediates, and it primarily occurs within the liver, the kidney, the heart. And the electrons from NADH in the cytoplasm are transferred to complex 1. And so we maintain that 2.5 ATP per NADH with this particular shuttle. So let's try to make sense of this one. Here's our NADH within the intermembrane space. So NADH can make it through the outer membrane of the mitochondria like most things, but it can't make it through the inner mitochondrial membrane. And so here's how it works. Those electrons ride on oxaloacetate to become malate, right? We've seen this reaction before many times. Okay, so we've used NADH to reduce oxaloacetate into malate. And there is a malate alpha-ketoglutarate transporter. It's a transporter that allows both malate and alpha-ketoglutarate through it. So malate goes through it, and there it is reoxidized back into oxaloacetate, and we get our NADH, which then can go and donates electrons to complex 1. However, what we've also done here is the net effect is to move oxaloacetate from the cytoplasm into the matrix. We don't want that. Shuttles should have one effect, moving NADH from one place to another. That's it, okay? One effect. So that means anything that was done to move NADH here has to be undone. So we need to somehow get oxaloacetate back here. Well, we can't obviously go backwards because we would be taking the NADH with us. So the next step then is oxaloacetate gets aminated from glutamate. Okay, so glutamate is an amino acid. It's going to donate an amino group to the oxaloacetate. And in so doing, oxaloacetate becomes aspartate. This is necessary because oxaloacetate doesn't have a way to get straight through the inner membrane. So instead, we use glutamate to aminate oxaloacetate into aspartate. And in so doing, glutamate becomes alpha-ketoglutarate. Okay, so now we have aspartate, and look at this. We've got an aspartate transporter right here, and it manages to get back. Okay, and then aspartate is reconverted into oxaloacetate. Its amino group given back to alpha-ketoglutarate to make glutamate. Okay, well, we're almost there. But the problem is we have just moved an amino group from here within the matrix to here in the intermembrane space. And again, we want the net effect to be just the movement of one NADH. So anything we do, we have to undo in order to get NADH there, which means we somehow have to get that glutamate, that amino group back here. So how does that work? Glutamate carrying that amino group moves through the glutamate aspartate transporter back here. And the alpha-ketoglutarate, right, where we had the amino group taken from, that trades places and goes through the malate alpha-ketoglutarate transport out here. Now we are at a net of NADH moving from the intermembrane space to the matrix. You will not be responsible for memorizing the enzymes involved in this. However, you do have to know where those electrons are riding. They're riding on oxaloacetate to become malate, and then back to oxaloacetate. And then the amino groups are riding on oxaloacetate to become aspartate. Now we're going to be talking about how amino acids are made and how amino acids are broke down. And so you're going to see these relationships again anyway, so you may as well memorize it now. Where oxaloacetate and aspartate, the only difference between them is an amino group. Glutamate, alpha-ketoglutarate, the only difference between them is an amino group. Okay? So you will have to memorize that aspect of this. And now we are back and we know the answer. What is it? Does the oxidation of glucose make 30 ATP or 32 ATP? And the answer is, it depends upon what tissue you're talking about, and therefore what shuttle is being used to move the NADH in the cytoplasm to the matrix.